Good morning, good afternoon, and good evening, everyone. And welcome to the Scientist.com Inside Scientific and Crown Bioscience webinar titled Advanced In Vitro Technologies to Investigate Therapeutic Impact on the Cancer Immunity Cycle. I'm Liam Sanyo, and I'm very pleased to be your host for today's webinar. From the Crown Bio team, we're being joined by Dr. Rajendra or Jen Kumari, Executive Director of Integrated Solutions, and Dr. Gera Goverse, Director of Immuno-oncology. Drs. Kumari and Goverse will be giving an overview of ex vivo and in vitro methods within the cancer immunity cycle framework and discuss the advantages and limitations of different model systems and how they can inform the decision to progress to in vivo studies and the clinic. And without any further ado, I'm very pleased to welcome the first speaker, Dr. Jen Kumari. Jen, thanks so much for joining us today, and the floor is yours whenever you're ready.
Thanks, Liam, for the introduction, and thank you to everyone for joining today's webinar. So today's webinar will be in two parts. I will overview the cancer immunity cycle to look at the regulation, impairment, and therapeutic intervention, and then Gera will focus on the different technology platforms, which will include traditional in vitro immuno-oncology assays and more recent advances in vitro technologies that can be applied to interrogate the cancer immunity cycle and therapeutic investigations. So there are multiple investigational strategies aimed at enhancing the anti-tumor immunity and overcoming the immunosuppressive pathways within the tumor microenvironment, which aim to protect those tumors. So this is nicely illustrated here. So although immune checkpoint blockade has been a breakthrough for cancer immunotherapies, the majority of the patients don't achieve a durable response. So alternative strategies are being pursued, looking at other checkpoint blockades or beyond checkpoint inhibition, including adoptive cell therapies targeting the tumor directly. You have vaccines to stimulate the innate and adaptive anti-tumor responses, or targeting the tumor microenvironment, and so on. And the aim with these strategies is to enhance T-cell activity, the trafficking to the tumor, releasing the T-cell breaks and changing the immune cell profile to increase immunogenicity. And we'll look at these strategies in different ways. So firstly, there is an unprecedented number of investigational drugs in immuno-oncology R&D, which is shown in this chart, documenting the number of active immuno-oncology agents for different immune therapeutic types, which is listed on the right, over a period of 4 years. The grey shows the agents in preclinical R&D stages, and then the clinical phases are shown in the different colours, and then the improved agents are highlighted in the yellow at the tips of these bars. So the large number of cellular therapies, the immune modulators that are not targeting T cells, vaccines, and then the T cell targeted immune modulators dominate the space and have grown over those 4-year period. So with the increasing number of investigational drugs, there's also an increasing number of clinical trials. So let's look at this next. So this slide focuses on just on the clinical trials for PD-1, PD-L1 blockade and the numerous combination trials. So here on the left, the number of clinical trials built on the anti-PD-1 and the anti-PD-L1. And so this shows that the collective number has increased from over 1,500 to over 3,000 in a period of 2 years. And this is dominated by pembrolizumab and nivolumab. And the majority of these are combinations with other immunotherapeutics shown in orange, targeted agents in teal, and then chemo in yellow, and so on. And then on the right, the number of trials is reflected is relative to the size of the bubble, with the largest number of trials for chemo combinations here in yellow, and then CTLA-4 combinations being the largest for the immunotherapeutic category. There's also LAG-3 and IDO shown here as well. And then you have targeted agents combinations such as VEGF, which is similar to the size of radiotherapy combinations going on, and then also other targeted agents such as PARP, EGFR, HDAC, and so on. So the aim of these trials is to achieve a durable response and survival, increased survival for patients, for a larger proportion of patients actually than currently seen, or at least identify the right patients for the right combinations that can achieve an effective anti-tumor immunity response. So in 2013, Daniel Chen and Ira Melman introduced the cancer immunity cycle, where they described the necessary steps needed for an effective anti-tumor immunity response. So if we walk through this cancer immunity cycle for those who may be unfamiliar, and this describes a series of sequential biological steps that need to be fulfilled prior to a successful or the successful immunological elimination of cancer. And the unique genetic alterations that are central to the oncogenic process can also lead to cancer cells appearing to be foreign to the immune system. And this is the first step in this cycle. So upon the death of cancer cells, antigens are released from the tumors. Now, most antigens are self-antigens, so tumors with high tumor mutational burden or microsatellite instability produce a better anti-tumor immunity response. And next, the antigens are taken up by antigen-presenting cells, resulting in, for example, dendritic cell priming, and then transported to the lymph nodes via the lymphatics. And here the antigen is presented to naive T cells within the lymph nodes, which results in T cell activation. And these T cells then migrate or traffic to the tumor site via the blood and infiltrate the tumor. And there's recognition of those tumors as foreign and eventually leading to T cell mediated tumor killing. And then further release of more antigens, and then the whole cycle starts again, leading to the eventual tumor eradication. So this is a very simplistic view of a highly complex system. And the cycle is actually it's coupled with a multitude of fail-safes, negative feedback loops, checkpoints that enable both precision and control, and also the ability to disengage and shut down the immune response. And this will then, just in case, to avoid damage to normal healthy tissue, or that is, self tissue, and avoid autoimmunity. So this is regulated by many different stimulatory and inhibitory factors to ensure eradication is precise and efficient. So now on this diagram, both the relevant stimulatory and inhibitory factors or biological process have been overlaid, and you can see the immune checkpoint regulators like PD-1, PD-L1 axis, the CTL4. You can also see the pro-inflammatory cytokines, so interleukin-1, TNF-alpha, co-stimulated factors like CD27, OX40, and so on. And also these regulators have different activities in different steps. So, for example, the T cell receptors are activated within the lymph nodes, but then they recognize the antigen within the tumor microenvironment. But in a cancer patient, the cycle might be impaired at any one of these steps, and this will result in an ineffective anti-tumor immunity, allowing that cancer to develop and progress. So we can now overlay how the cycle can be impaired. So firstly, you get impaired release of antigens, which typically would have been via the immunogenic cell death process. And this can occur in the absence of dying tumor cells and would lead to impairment of subsequent steps downstream, which involves antigen presentation and also T cell activation. So restricted antigen presentation and T cell recognition can also be associated with mutations in the HLA or loss of HL alleles. And then the factors within the blood, like cytokines and systemic inflammation, can impact the activated T cells, leading to T cell exhaustion. The influx of T cells can be impaired this is due to the composition of the vasculature and the cytokine gradient. And then once in the tumor microenvironment, the activated T cells face many immunosuppressive factors, preventing the T cells from functioning or doing their jobs. And this includes checkpoints such as PD-L1, LAG3, TIM3, immune modulating factors like IDO, IL-6, TGF-beta, or regulatory T cells, CAFs, macrophages, myeloid-derived suppressor cells within that tumor microenvironment. And ultimately, this leads to impaired tumor killing. So to repair these blocks in the cancer immunity cycle, various immunotherapeutics have an opportunity to intervene. And there are numerous targets due to this complexity of the cancer immunity cycle. And as a result, this presents a number of different therapeutic opportunities. To give an example, the potential targets for immunotherapeutics are now overlaid this time to try and, so what they're doing are trying to stimulate or reignite the cycle. And these are referring to agents that are actually in clinical trials at the minute, as well as those that are already approved. So this is looking quite complex, and you can take up the different layers just to simplify it. But this does actually reflect the huge number of new drugs that are being developed that I mentioned earlier, as well as those clinical trials that are ongoing and the different types of agents involved. So here at the first step, these therapies like the targeted agents, so you've got the HDAC inhibitors, PARP inhibitors, you've also got oncolytic viruses and the standard sort of therapies like chemo and radiotherapy. They're all killing the tumor to ignite the cycle by releasing those tumor-associated antigens and potentially inducing an abscopal effect. Or you have the vaccination approach to boost antigen presentation. There are a number of different approaches to boosting T-cell activation, such as immune checkpoint inhibitors. You've also got cellular therapies down here, which also impact the T-cell priming up here, as well as the bi and tri-specific T-cell engagers or killer cell engagers. And then you also have the cytokine and co-stimulatory factors involved in the T-cell priming and activation, which are in clinical trials at the minute. And then within the tumor microenvironment, you have a range of immune checkpoint inhibitors already available, and these are hugely successful. And this really is the final step in the process. So it's important to kind of kickstart the killing of the tumor, which is why these agents are so successful. But what this also highlights is how the different stages or any of these stages and impairment in any of these stages can actually reduce the impact on these immune checkpoint inhibitors or this final step, and that the suppressive tumor microenvironment also has a major impact on that final step. So with this level of complexity, we have to ensure that our preclinical models and tools are able to dissect the biology of the cancer immunity cycle to answer specific questions or hypotheses on immunotherapeutics and immuno-oncology targets. So first, I'm going to quickly overview the in vivo tools to study the cancer immunity cycle. And this can be divided into either murine systems to assess mouse analogs such as syngeneic models or gem models, or human-specific systems where the mouse analog or the cross species is not available. So the human target is needed, and this is achieved either through reconstitution of the immune system or engineering in a human target. So there's also publications available to refer to, which is shown here, as well as numerous resources on our website, including databases on our models. And so this first publication shows the animal models for immuno-oncology and is a good overall guide on the strengths and limitations and the properties, as well as the utility of the different animal models. Again, this is summarized in this table. And so I'm just going to focus on the immune competent systems such as syngeneics, because these are widely used for immunotherapeutic evaluation, including many of the ones discussed like immune checkpoint inhibitors, as the whole cancer immunity cycle can be modeled in these animals. So these models are robust, they're also easy to use, but they do lack clinical relevance, and it is murine biology, and this it doesn't accurately reflect the human immune system. So one of the most commonly used cancer cells here is MC38. And this does actually have a high mutational load, which reflects the hypermutated or the microsatellite instability seen in colorectal cancer. But these mutations are not the same as that seen in humans. And this could be due to the fact that these are carcinogen-derived models, and also they've adapted to grow in 2D culture over many pathologies. And these models are typically implanted subcutaneously, but you can model the complex tumor environment by implanting the cells orthotopically, which can actually be more time consuming. So overall, these models can take time to develop, can be costly. And also, so what we did in the second paper was actually to show and highlight the options for both in vitro and in vivo, looking specifically for CAR T therapies, which again just highlights the advantages and disadvantages of each of the systems. But for this series of webinars, we wanted to focus on the in vitro assays, as these can be more flexible, adaptable, and scalable, and used to interrogate immunotherapeutics and combinations in a step-by-step manner with the cancer's immunity cycle in mind before moving in vivo. So overlaid now on the cancer immunity cycle, you can see a variety of different assays that are offered by Crown Bioscience, studying the different steps. And these assays include both the traditional and very novel assays, including organoids, 3D systems, high content imaging, and ex vivo platforms. And you'll be hearing more about these in the series of webinars. And with that, I would like to thank everyone for their attention. I'll now hand over to my colleague Gera for the second part of today's webinar on the different in vitro technologies.
Many thanks, Jen, for this great introduction on the cancer immunity cycle, explaining the different steps and the possible therapeutic targets to stimulate immune cell-mediated killing of the cancer. Within the second part of today's webinar, I will present on the different technology platforms to analyze the cancer immunity cycle. First, I will give a quick overview on traditional assays that are already longer available, which you might be more familiar with, followed by focusing on the advanced in vitro assays that have been developed in the last decade. Again, within this cartoon on the cancer immunity cycle, you can already find a summary of those assays offered by Crown Bioscience, studying those different steps, which actually show both the variety of traditional and very novel assays. As already introduced by Jen, different in vivo models could be used to study the cancer immunity cycle. While those might contain a complex tumor architecture, including vasculature and an immune system, depending on the model, these in vivo models often show a lower scalability and are involving in a murine system. Whereas on the left-hand side of this slide, we can find a traditional assays with 2D cultures, which have a more simple setup and thereby would allow high throughput screening with well-defined cells, but on the other hand lack morphological characteristics and complex cell-cell and cell environmental interactions, with lower translation to the clinics. Thereby, these traditional assays are on two ends of the balance of scalability and physiological relevance. First, I would like to discuss different technology platforms that could be used in both of these traditional assays, as was nicely listed by Yuki et al. In this paper from 2020. And from these different technology platforms, I will quickly share with you some relevant examples from literature that made use of these technologies. For example, within this paper of James Ellison's group, where they have treated mice with P60 melanoma tumors with different ICI therapies, targeting both the CTLA-4 and PD-1 pathway. This nicely demonstrates a strong increase in infiltration of tumor factor CD8 T cells by FACS analysis. Additional combination treatments were detected and similar results, but to a lesser extent, were observed with effector CD4 T cells. On the other hand, the number of regulatory T cells within the tumors were reduced. Another example nicely showed the use of single-cell analysis, which can be performed with different technologies such as RNA sequencing. Within this paper from T. Roche et al., they have analyzed the single-cell expression patterns of more than 2,000 T cells from multiple patients with melanoma. They identified T cells and their main subsets on the basis of the expression levels of their respective defining surface markers. Additional principal component analysis distinguished the T cell subset and showed their heterogeneity of activation status on the basis of the expression of both naive and cytotoxic T cell genes. In this example from the Rosenberg group, where they studied the effects of TCR products, they were able to show that indeed engineered T cells for specific tumor antigens were able to secrete high levels of interferon gamma when they were co-cultured with T2 cells that were specifically pulsed for those tumor antigens. And these studies further supported the development of such cellular therapies as TCR products. As last example, I would like to give you I would like to share with you this data on the T cell cytotoxic killing effects that were measured in studies where they wanted to, in the end, had the goal to actually look for the relevant role of CD3 expression for the design in T cell engagers. And in this case, they analyzed different cell lines that actually had an overexpression of this anti-CD3. And in those different cell lines, when they indeed show this CD3 overexpression, you can see within the first row, which was labeled with A, that there was a reduction measured of live cells via viability dye. In addition to that, in the second or in the middle row, you can actually see that besides the reduction of live T cells, indeed, there was an increasement of annexin 5 positive cells. And in the lowest row, you can find that that also is aligned with an increase in interferon gamma production in these co-cultures. So all those different technology platforms, they could be of use to actually, in the end, select a drug and make sure that those drugs that demonstrate actually high sensitivity or show actually promising results go into clinical trials. And as was already nicely shown by Jen, there are actually the increasement of the clinical trials for immuno-oncology drugs is increasing over the last years. However, we also know that there's still a lot of clinical failure. So although a lot of clinical trials are started, not all of them will end up to be successfully. And part of that reason for the failure of those clinical trials is because of the lack of relevant preclinical assays. And we think that actually both a complex tumor microenvironment that includes different cellular players, but also relevant expression levels and making use of the human immune system playing an important role to make sure that preclinical testing has been done in a correct way. And therefore, we think that 3D cell cultures could could actually close the gap between preclinical testing and clinical testing, whereby those different points can be included. And therefore, I would like to share with you some data. In this example, they actually compared 6 different breast cancer cell lines that were grown in both 2D and 3D, which were treated with chemotherapeutic agents. And already within the images on the slide, you can see that there are, of course, morphological differences when the same cell lines were grown in 3D. And if they were looking at the drug sensitivity, which is shown on the graphs within the right end of the figure, you can actually observe that upon treatment with 5FU, similar sensitivity was detected between the 2D and 3D cultured cells. However, if we look specifically on those 3 cell lines that are highlighted right now, which actually showed also a more dense spheroid structure compared to the other cell lines, you can actually observe differences in sensitivity to the treatment with either plexitaxel or doxorubicin compared to the 2D cultures. And this is a first example actually of showing differences in the sensitivity of cell lines that were either cultured in 2D or 3D. In another example, they looked not only at the sensitivity of the treatment, but they also looked at the expression levels within cell line DLD1 colon cancer cells that were grown in 2D and 3D. And they were studying specific inhibitors on the ACT-mTOR S6K pathway, and therefore they also stained for the expression of phospho-RP-S6. And already within the control conditions upon treatment with DMSO, you can observe in both 2D and 3D cultured cells that they had a nice expression of this phospho-RP-S6. However, although in 2D culture all the cells were stained to equal intensity, you can see that in the 3D cultured cells in the spheroids, the expression on the outside seems to be more intensive compared to the expression within the inner side of the spheroid. However, this staining pattern seems to be quite equal from high expression in the outside to a low expression in the inside. If we then look at the first treatment with rapamycin and thorium-1, you can appreciate the similarities between the 2D and 3D cultured cells, whereby actually the cells are being killed, and in addition of that, there's no expression anymore of phospho RPS6. If we then look at the 3 remaining treatment conditions, where you can actually, if we just first focus on the PF compound, you see a reduction in the phospho expression in 2D, which is also observed in 3D. But you can also see that the pattern has changed. And this actually becomes even more clear in the last 2 compounds, where actually the expression levels of the phospho seems not to be affected in 2D cultures, but only in the 3D cultured cells. So here you can clearly see that although there's still expression detected, the pattern is completely changed compared to the DMSO control. You can appreciate that it's a little bit more scattered compared to the control conditions. In addition, I would also like to highlight the role of the extracellular matrix in 3D cultures. And this extracellular matrix allows not only to have cell-cell interactions, but also to have interactions of the cells with this extracellular matrix. And thereby that could actually influence the cells in their migration and growth, and actually their growth into these organotypic tissues. And a nice example for that is shown on the right-hand side, whereby a breast cancer cell line which was grown at Crown Bioscience, either in an ultra-low attachment plate at the bottom, or in this 3D matrix embedded that does include extracellular matrix on the top. And you can clearly appreciate the differences. So whereas in the bottom in the ULA plates, the morphology is really round, but actually only within this 3D cultures that do contain extracellular matrix, we observed a branched phenotype or an invasive phenotype of the tumor model, which was clearly absent in those ULA plates. So first of all, show some data why these 3D cultures can be so valuable in preclinical testing. So in this example, they performed co-culture of cancer cells together with fibroblasts and immune cells. And what was very interesting that in these studies, they were able to analyze also the infiltration of immune cells. And they specifically used antibodies for IL-2 variant, and they indeed were able to show that upon treatment with these antibodies, that both in immunohistochemistry, but also after analysis of the numbers, that both CD45 and CD3 cells were increased. And if we look at the bottom half of this graph, you can also appreciate that both the tumor volume was reduced, but also the CEA expression of the tumors was reduced. And in specifically this study, they nicely followed up these studies after these IL-2 variant antibody treatments with bispecifics for T cells that were either targeting tumor antigens or fibroblasts, and this could then further enhance actually immune cell infiltration into those 3D tumor structures. So this brings me back to this balance that I previously showed, whereby those 2D cell cultures are on the left-hand side, so they're very high scalable, but however they might be a little bit less physiological relevant. And I already showed some data to you about the use of 3D cultures with cell lines, so making use of spheroids would already have a much higher physiological relevance. But I will also follow up in the coming slides on the use of organoids, which have a more are patient-derived and thereby have an even higher physiological relevance, but also the inclusion of ex vivo patient tissues in 3D cultures. And what's important to remember that although they are more on the right-hand side of this specific balance, as they do have a much higher physiological relevance, these models are also very well scalable, and especially if we compared it with in vivo models, maybe somewhat less than 2D cultures, but also that depends even on your systems that you have available. So first of all, some background on the organoids. So this technology was first described by Claver's lab with colorectal cancer organoids, whereby they were able to isolate LGR5 positive stem cells from the crypt of the intestines. And by culturing those stem cells into these 3D cultures, they were able to let those stem cells grow into organotypic tissues, which were described as the minicuts and are known as the organoids, with different cellular layers of the epithelial layer, as you can find them in the patient. So those organoids have a much more heterogeneous population of different cells that are derived from the stem cells. And as you can see within these images on the bottom, you can also appreciate that different mutations might lead to differences in the organoid, in how their morphology is looking like, especially compared to a healthy colon organoid. As you can see on the right hand side, you can appreciate that those colon cancer organoids shows often a loss of lumen formation or a different multiple lumens in one organoid, but also the epithelial barrier, it can be much more thickened. And how these organoids can be used in a screening type of assay was actually nicely demonstrated by ourselves in a collaboration with our partners in the Suppress STEM project. First of all, in this project, 3 patient organoids were screened for more than 500 specific bispecific antibodies. And from those antibodies, we made a hit selection based upon whether we detected a certain amount of inhibition on the organoids compared to the two other compounds. And from those hit compounds that were identified in the first screen, we followed up with a bigger screen that included more than 30 colorectal cancer organoids. And then indeed, from those hit compounds, we identified specific hits that showed a broader effect on all those different patients. And as you can see within this graph, where you can see the black line, and if you compare that, for example, with other antibodies targeting also the EGFR pathway, like cetuximab, the effect on the tumor organoids is much stronger. In addition to that, in these studies, there was also a nice comparison made between both tumor-derived organoids from one patient, but also including normal-derived organoids from the same patient. And it also nicely demonstrated that only the tumor organoids showed sensitivity and not the normal organoids. And this is just one example that we recently were able to publish in Nature Cancer of this year, nicely demonstrating the use of this organoid, but also the high content imaging platform, which I will further introduce at a later stage of this presentation, which actually led that this molecule was brought to clinical trials by 2018. So how can those organoids be made use of? So this is a literature example. In this case, they actually performed a live imaging technology. They made use of colorectal cancer organoids, and they were expressing GFP signal, and they were co-cultured either with the parental NK cell line or EpCAM CAR NK cells that were generated or were genetically modified from the parental cell line. And as you can see within the graph already quite nicely, is that there might be already some effect with the parental NK cell line. However, you can clearly see upon time a reduction of the organoid positive area if they were co-cultured with these specific EpCAM CAR NK cells. And this is also shown within the images on the right and in the figure, whereby you can clearly see that after 10 hours treatment or co-culture with these EpCAM CAR NK cells reduced the GFP pattern of the organoids, but also showed a real high infiltration of those NK cells in the organoid tissue. Previously, I would like to further give a more in-depth introduction on the high content imaging platform that was already used within this Nature Cancer paper. So all the different type of assays, so 2D or 3D cell cultures, can be used in this platform. And what this platform is shown within this figure, so there's a culture in 384 well plates of the tumor tissue of interest, and they will be co-cultures with cellular players of interest together potentially with compounds, which will be followed with the high content imaging platform, which is able to reconstitute and score the images in 3D. And how does that look like within the images? So in this high content imaging platform of Crown Bioscience, we are actually staining actin positive cells, the nuclei with TAPI signal, and in this case T cells. They were labeled before they were added to these cultures, and they can be detected in green. And from those raw images, you can see that we are able to actually extract individual organoid objects, but also individual immune cell objects. And from those different segmentations, we can measure both readouts on the organoids, but also on the immune cells. For example, whether organoid volumes are reduced in a certain co-culture condition, but also whether immune cells are proliferated or analyzing their spatial relation towards the organoids. And this becomes very interesting when we start to overlay those two different segmentations. Then we can actually analyze the infiltration of those immune cells into the organoids. And this is also nicely visualized in this higher magnification, a rotating image, whereby you can see that some of those immune cells really start to infiltrate into an organoid. And this can be analyzed with this high content imaging platform. So with this high content imaging platform, we can first of all analyze the migration of immune cells. As you can see within the video on this slide, which is actually a side projection of a well, you can see the tumor tissue in blue, which is rather more on the bottom located. In addition to that, you can see that immune cells which were labeled in green, which are deposited on the top of this hydrogel mixture. From the beginning of the move, you can see they start to migrate towards the tumor tissue upon time. And this is something that we can measure and analyze. For example, certain treatments seem to enhance this migration of immune cells. And this can be analyzed within a graph when we actually plotted on one end the set position of the well, and on the other hand, the total immune cell count. And here you can clearly observe that indeed a certain treatment led to a higher peak of immune cells at a lower set position, more closely located to the organoid or tumor tissue compared to the untreated control conditions. In addition to this migration, we can also analyze the infiltration and the subsequent killing of those tumor by the immune cells. And again, within the video, you can see that at the beginning, the immune cells in green start to aggregate and infiltrate the tumor. And at the end of the movie, you can see that the tumor is almost completely killed. To further highlight this process and those steps that go hand in hand with each other, I would like to share with you some data whereas in this plot we have plotted on the y-axis the total tumoroid volume, and on the x-axis the number of infiltrating T cells in this case. So we can see within tumor-only conditions, there's no killing detected, so we have a high total tumoroid volume. We see that if we add T cells, there is on one hand a reduction of volume, but also in this time point we still detect quite some infiltration of the T cells. This the killing effect of those T cells can be measured upon a specific treatment, so it reduces the total tumoroid volume. And if we look at a later time point, we actually start to see that there's even a much stronger killing observed of those T cells. As you can see that the plot is almost in the bottom left corner. And you could also nicely appreciate that there's actually kind of movement ongoing. So in the conditions when there's no killing and no infiltration on the left top hand, then there's a intermediate phase of intermediate killing and some infiltration. But at the end of a culture, when there's very strong killing by treatment or activation of the immune cells, You can see them appearing completely in the left bottom. I also would like to give you another example from the possibilities with our high content imaging platform. So in this case, we performed co-cultures of 5 different colorectal cancer organoid models together with PBMCs, either in the absence or presence of superantigen treatment. And you can nicely appreciate that within those 5 different colorectal cancer organoid models, there's differences in sensitivity towards the addition of immune cells. Whereas the addition of just PBMCs in most of the models only slightly reduce the total organoid volume, you can see that upon addition of superantigen, this reduction is clearly enhanced. However, some models show more sensitivity, whereas other models show a higher resistance towards the killing of those immune cells. Of course, there can be different reasons for that. That can be either on the tumor microenvironment or specific genetic expressions of those organoids, but also on the morphology. And I would like to highlight that actually within the second webinar of this series, we would further continue on explaining these type of assays and how you can make use of them to address your compounds in vitro assays to test within the cancer immunity cycle. Another example that I would like to share with you, which was nicely published this year in Nature Biotechnology by Dekkers et al., is where they generated the technology that they call Behaved, which was allowing them to track immune cells via imaging-based technology analyzing their behavior actually of those engineered T cells. And as you can see, within specific co-cultures with organoids that either had or didn't have expression for in the top left graph for HLA-A2 or for WAR1, which is shown in B. And if they were added with either those specific T cells that could recognize those tumor antigens, you could either see an enhancement in killing, but in the absence, nothing was detected. And this is also nicely shown within those images. However, in addition to that, they were nicely able to track the killing of those engineered T cells on specific organoids, they were also able to analyze the behavior of and the movement of those engineered T cells. And thereby they were able to show that actually the CD8 tags had a preferred super engager type of movement, so they were very mobile, whereas the CD4s really showed that they had a lower mobility and showing more what they called lazy or slow scanner type of characteristics. In addition to the analysis of the T cell capabilities in co-cultures with organoids, these assays can also be, these co-culture assays can become more complex by incorporating more cellular players and thereby try to reconstitute the tumor microenvironment. For example, in this specific example from Crown Bioscience, we incorporated not only a co-culture with T cells, but also with monocytes. And as you can see within the graph, whereas pre-activated CD8 T cells have a high capacity of infiltration into a tumor object, when we added CD14 positive monocytes, you can appreciate that there is a reduction of those immune cells that are infiltrated in the tumor tissue, really showing a suppressive effect of these myeloid subcellular players. And these effects can, on the other hand, be partially rescued upon treatment with specific drug compounds that were targeting the polarization of these myeloid cells. So as I just gave for a couple of examples, we can reconstitute the tumor microenvironment by incorporating both tumor tissues, for example, organoids, which are coming from the patients, together with different cellular players, both coming from the immune cells, so for example T cells and myeloid cells, but also combine them with cells from the stroma compartment, like the fibroblasts. And those co-cultures could be analyzed, could be used as an assay to analyze the effect of specific drug therapies that would target the cancer immunity cycle. However, in addition to this reconstituted type of assays, there's also the availability of working with the native tumor microenvironment. And although there's also in these type of assays we will be working with patient material as well, it means that there's no pre-culture stages, for example, to generate organoids, because by generation of the organoids, you might also lose, for example, your tumor-infiltrating lymphocytes or other cellular components that are present within this native tumor microenvironment. However, both of these two different types of assays, so as well as the reconstituted as the native tumor microenvironment, can be analyzed within the same type of readout in these 3D cultures. For example, with our high-content imaging platform, but also via cytotoxic analysis, cytokine profiling, or FACS. So first of all, I would like to give another example from the literature where they actually made use of what they call patient-derived organotypic tumor spheroids, also known as the PDOTs. They analyzed these PDOTs in a microfluidic system. First of all, they analyzed and phenotyped the immune cells that were available in different patient materials. So they tested a lot of patient samples. And indeed, within those different samples from different indications, they were able to detect differences in the immunophenotypes in those patients. In addition to the analysis, they also performed immunohistochemistry stainings or immunofluorescence stainings on those 3D cultures. And indeed, they could confirm differences in morphology, but also differences in the number of infiltrating immune cells as shown in these images. Subsequently, they were able to perform treatment with different immune checkpoint inhibitors. And for example, in this example, it's shown for upon treatment with anti-PD-1. And they were able to analyze different cytokine profiles of those different patients. And they actually show overall within all those patients an interesting upregulation of CXCL13 and CCL19 upon treatment with anti-PD-1. Then I would like to end with at least one data slide from one of our technologies or assays available at Crown Bioscience ourselves. So this is an ex vivo patient platform where indeed the ex vivo material from the patients is directly cultured in our hydrogel platform, and then a high content imaging analysis is performed. And these high content imaging analysis, as you can see, could nicely analyze, for example, the tumor size, but also analyze immune cell proliferation and combine those results with the analysis of intergamma cytokine release. And this is just one example within all the different patients that we have tested. Different immune monitoring drugs have been tested, like immune checkpoint inhibitors. But there I would also like to note that in the third webinar of this series, we will follow up in much more detail on this specific platform. Yeah, it would be too much to explain on one slide. With that, I would like to again show a summary slide showing different technologies with both traditional and novel analysis platforms that can be used for the in vitro testing of the cancer immunity therapies, but also allows testing of combination therapies. However, the in vitro setup is very important to make sure your data will be physiological relevant, and making use of either, for example, organoid 3D cell cultures or ex vivo patient material that can be grown in 3D, including a native tumor microenvironment, will be very valuable in your preclinical testing to have higher chances on clinical successes. So thanks everyone for listening to our presentation. We now will be taking your questions in a live Q&A.
Fantastic. All right. Well, thanks so much, Gera and Jen, for the great presentations. All right. Well, let's jump in with a great question here. So which type of assays will allow for the analysis of immune cell migration or infiltration?
Yeah, thanks, Liam. I think that's indeed a really good question. So actually, to be able to measure immune cell migration and infiltration, the best technology that can be used is probably using the imaging type of technology. There's, of course, alternatives available. For example, working with if you have two different compartments, and then you can also be able to measure the migration or movement of cells. However, that would not be in a 3D type of assay. So really the imaging platform, I think, is the only one that can measure migration and infiltration of cells, which I think has nicely been shown in literature, available assays, but also available at Crown.
Fantastic. Great answer. Next one here. So how translatable are the in vitro models compared to some of the in vivo models mentioned for checkpoint inhibitors?
Yeah, I can answer that one. So I think generally the most translatable models are the ones using the patient-derived material. So like the organoids, like the EVPT, and then for in vivo, the patient-derived xenografts. And these really do represent the heterogeneity and the genomic aberrations you see in the clinic. But the thing is, when you are using in vivo, the PDX models, you have to reconstitute the immune system into immunodeficient animals. And this is typically done using donor PBMCs from healthy patients or CD34 positive hematopoietic progenitor cells. And you can achieve reasonable development of the CD4 positive and CD8 positive T cells for immune checkpoint evaluation. And similarly for the organoid co-cultures, you can use donor PBMCs. And these, again, can be used for those immune checkpoint inhibitors. I think there are limitations in that you are typically, if you look at the cancer immunity cycle, only mimicking the second half of that full cycle. And this is because of the lack of T cell priming or the activation following antigen presentation. And ideally, you can actually use matched autologous systems, and this would avoid any alloreactive sort of response. So I think, as you heard about the autologous organoid systems, have a great advantage of being not only relevant, scalable, but also more relevant for something like the immune checkpoint inhibitors. And again, you have the EVPT, which again has endogenous immune cells as well as stromal cells. So again, they're representing that tumor microenvironment more faithfully. So these are really very highly translatable models.
Yeah, great point. Thanks, Jen. Another nice question here. Can patient-derived organoids be used in vitro assays without the use of extracellular matrix?
Thanks, Liam. I will take that question. So I think in general, to get organoids growing, they need to be in an environment where they have this extracellular matrix, also for letting them grow into organotypic tissues. However, thereafter, you can take them out of this environment and maybe use a lower type of, a lower percentage of the extracellular matrix in assays. So thereby you have a bit more flexibility. However, I think we do believe that actually keeping this extracellular matrix is very valuable, specifically also to make sure that if you are going to make a complex co-culture by incorporating different cellular players, that thereby also still this 3D environment remains consistent there, and also immune cells or fibroblast cells will not just be in contact with organotypic tissues by coincidence or by gravity, but really by making sure that those cells move to each other. So it's, I think, much more relevant to use extracellular matrix in those type of assays.
Fantastic. All right, next question here. So from which type of indications can organoids be generated and used within in vitro testing?
Yes, I will take that one as well. I think that's also a good question, and it's important also that with all those different indications that are out there with cancer, that can we all address them in this type of assays? So definitely, actually, the organoid technology is extensively, and also within Crown, for example, we were able to generate organoid models from, for example, PDX models, so from our in vivo mice models that are available, which further extended our organoid portfolio available, and thereby also really increasing the type of indications which were maybe still limited in the organoid field. So actually, many indications that you can come up with are available in organoid technology, and thereby making them really a successful type of in vitro assay to use for your testing.
Excellent. All right. I think in the interest of time, we'll just make this next question the last one. I think it's a great question to end with. Do you think 3D model systems will replace in vivo models?
Yeah, I think in some cases, maybe especially where there's no relevant in vivo model, then yes, 3D models may be the only option. And I'm specifically thinking about where there's early stage cancers which you can't develop in vivo models, but you can certainly develop organoid models. But I think in the majority of cases, 3D models will be necessary for maybe refining some of the in vivo experiments that are conducted, and also how and when we enter those animal studies, because those animal studies can be very time-consuming, laborious, and expensive. And also, I mean, there is still a requirement for in vivo systems, especially if you're looking at the impact of that, again, that whole cancer immunity cycle. So, for example, and another case is if you're looking for combinations, let's say, to move forward with in vivo, I think in vitro can really screen out those potential combinations. Whereas in vivo may be more advantageous if you're looking at a specific tumor microenvironment like the brain or the bone, or generally just metastasis. So the selection, I think, of that appropriate model system is going to be based on the specific question is being asked, that will ultimately determine which model system is going to be sufficient. And if you look back at some of the combination strategies for ipilimumab and nivolumab, the MC38 model was used extensively in vivo, whereas in vitro the assays were looking at showed enhanced cytokine secretion. So there is a combination of the two types of systems really kind of building the whole story when you are looking at some of those maybe combination strategies.
Excellent. Yeah, some great points about the availability and appropriateness of models. Well, Jen and Gera, thanks so much for joining us today. It's really been a pleasure having you with us.
Thank you.
Thank you so much.
So in closing, we hope you enjoyed this Crown Bioscience Scientist.com and Inside Scientific webinar, and we hope to see you at the next one. Have a great day, everyone.